Now we are going to discuss about the co-transduction mapping. So the co-transduction mapping, what happens? There is a maximum size of the DNA can be packed into the bacteriophage during the transduction. P1 can pack with 2 to 2.5% of the E. coli chromosome. P22 can pack with 1% of the Salmonella typhimurium chromosome. Co-transduction is referred to the packaging and transfer of two closely linked genes. Co means with, co with. So the co-transduction mapping is used to determine the order and distance of the genes, okay, that lie fairly close together. Co-transduction means simultaneous transfer of the two genes. Okay, you will understand about this example. Consider the example of following two E. coli strains. The donor strain will with the genotype arginine plus, methionine plus, and streptomycin sensitive. Then recipient strain with the genotype arginine minus, methionine minus, and streptomycin resistant. The steps involved into the co-transduction experiment like this. Donor DNA will have the arginine plus, methionine plus. These two genes are closer to each other. Okay. Hence, there is a possibility of the transfer of the these two genes because these are the closer genes. Okay. This into the phage DNA, there must be chances of packaging of the two DNA. Here you can see this is a P1 phase. Okay. Then P1 phase, it is a E. coli phase. So cells and phages are there. Phages will get adsorbed. They will penetrate their genome and they will lyse the cells. You can see here there the more number of the cells, here the less number of the cells. These recipient cells are arginine minus and methionine minus and streptomycin resistant cells. Okay, these are the recipient cells. And when these recipient cells are grown along with this lysate, this is the P1 lysate will contain an occasional phase will contain the piece of the bacterial chromosome. Okay, we can that in phase population, and then new flask containing the millions of the recipient cell, and recipient cell will have the arginine minus, methionine minus, and streptomycin resistant. Then new flask containing the millions of the recipient cell, and these cells will get infected with the this P1 phase, which may have the methionine plus and arginine plus gene. Then the plate onto the minimal agar plate containing the arginine and streptomycin, but without methionine. Here the 50 number of the cells will grow. It is the approximate number it is taken. The minimal plate with arginine and streptomycin means here the which cells will grow because donor cells are streptomycin sensitive. These donor cells will not grow here. The recipient cells are the streptomycin resistant. These will grow, but this for the growth of these types of the recipient cell will require both of the amino acid arginine and methionine. But here only arginine is present. Hence, the cells, these recipient cells will not grow. Arginine minus and methionine minus cells will not grow here. Only those cells will grow in which the methionine gene is transferred, means the methionine gene became the plus. Those cells will grow. And these cells, suppose there is a growth of the 50 colonies, then what they did, you can see here in next picture, these colonies must be of the methionine plus cells. To determine whether they are also arginine, stick onto the plate lacking both the amino acid, okay, so that we can identify whether these are the co-transform, means whether these recipient cell got both the genes or not, then pick up each of the 50 colonies and restrict them onto the onto the medium lack of both the amino acids. Okay, and those cells will grow here in which both the genes are transferred here in this case. And because of that, you can see here the growth of the cell obtained here. And because this genotype is the arginine plus and methionine plus, this genotype is also arginine plus and methionine plus. Okay, so that suppose in total number of the colonies obtained here the 21. Okay, and here how much among the how much colonies, number of the colonies obtained here the 21. Okay, and here how much among the how much colonies these are obtained, 50 colonies. Generally, then the co-transduction frequency can be determined by the 21 divided by the 50, that is the 0.42. So this is the 0.42 is the frequency of the co-transduction. Okay, means into the 100 cells, 0.42 cells were get transduced with the co-transduction. This is called as the co-transduction determination of the co-transduction frequency. And then co-transduction mapping by the scientist Wu in relation between the co-transduction frequency and map distance. It is obtained co-transduction frequency, that is the 1 minus, this is a standard formula, 1 minus d upon L brackets cube. D is the distance between two genes in minute. L is the size of the transduced DNA in minutes. For the P1 transduction, the size is near about 2% of the E. coli chromosome, which equals about 2 minutes. Okay, and then let's the equation of our example, we as we determine 0.42 is the frequency we determine equal to 1 minus d upon 2 brackets cube, because 2 it is the value of here like this, 2 is the value of the L, capital L. And in this way, after putting the values into the formula, we'll get the final answer b equal to 0.5 minutes. In this way, therefore, the distance between the methionine arginine genes is approximately 0.5 minutes. In this way, co-transduction mapping it is done. Okay, and this was